Good morning, everybody. I just wanted to welcome you back. Today what we're going to be doing is looking at the simple organization that we were talking about before of like 3 photoreceptors feeding into a single bipolar cell, and then that in turn passing on to a ganglion cell. And then we're going to elaborate on that and start to see how this type of approach can actually be used to help us be able to discriminate between stimuli that are being shown on the receptive field of the retina. So I've adapted some components that I normally would do in class to what we have here in online, and so we'll continue our bit of a hybrid experience until we get things and we see each other face to face on Thursday. We'll spend a little bit in the very beginning to get things organized and review and test ourselves, make sure that we're on point with some of these trickier concepts that are part of the visual system. Okay, so let's dig in. So we have light that's shining in, and this is a review briefly of where we were before, where light is shining onto, let's say, this particular type of photoreceptor, and then that's activating the biochemical cascade that's causing the closure of those sodium channels, which in turn hyperpolarizes this membrane potential of this photoreceptor, decreasing the amount of glutamate release from there. And as we've learned, glutamate is not always excitatory. As a neurotransmitter, what that means is that you sometimes can have receptors that are that see glutamate as being an inhibitory neurotransmitter, and others where you have receptors that are that see glutamate as being an excitatory neurotransmitter. And that's how these two types of bipolar cells are able to respond to the same signal of a lack of glutamate production here differently. And they respond differently. So less glutamate for this guy means less inhibition because it's not acting on that inhibitory receptor there. So then since the brakes are off, it's getting depolarized more readily and passing on neurotransmitter onto a ganglion cell here. And so in this simple chain, what we see here is that this on-center bipolar cell is then talking to a ganglion cell. That's an on-center ganglion cell, and that gets passed on to the LGN, et cetera, et cetera. And this is an example of an off-center bipolar cell that sees glutamate as being an excitatory neurotransmitter. Less glutamate being produced means that it's hyperpolarized relative to normal because there's less of this excitatory component being added to it. And so there's less glutamate being passed on here. And so this moves onwards. So typically we think of glutamate as being an excitatory neurotransmitter, but this is a key illustration of how neurotransmitters are sort of, they're not just built to be, they're agnostic in terms of whether they're actually excitatory or not excitatory. It's how they're received or how the receptor actually sees them. And that's what we're kind of seeing, this kind of difference in on-center and off-center bipolar cells. So an important point there about neurochemistry, and we're seeing it play out in systems biology. Okay, so then we talked about how light shining onto the surround here then influences the activity of the dark area here, the centered area here, through the action of horizontal cells. So this hyperpolarization of these particular photoreceptors here are in turn mean that we have less glutamate released, and that in turn helps to shape the responsiveness of the horizontal cells. Sometimes what you'll hear from physiologists is that they'll be like, oh yeah, that shapes the responsivenesses. And then you ask, well, what neurotransmitters right here? And sometimes they don't have the answer. Instead, what they've been able to do is characterize it by how the cells respond to each other, but they may not know the entire mechanism of the whole piece of it. And so in the references that I've been able to find on that, I have not been able to find a good, clear name of the neurotransmitter released here and the receptor here. But so that's what we have here. So let's take a look then and dig in and see a little bit more about how this setup works. We talked about inhibitory and excitatory glutamate receptors, so I'm going to move on from there. And what we're going to do now is look at how this is set up. And this is where we dropped off last time, describing how we have the photoreceptors that are getting activated, they act on bipolar cells here that in turn feed to ganglion cells. Unlike what we saw on the previous page, where there was a one-to-one correspondence between a bipolar cell here and a ganglion cell here, this is showing something interesting. It's making it and reflecting a little bit more of a realistic type of connection within the retina of how these different bipolar cells communicate with each other. So there's there's gap junctions that help to connect some of these bipolar cells to talk to each other, which is kind of interesting. I think the other thing is that an on-center ganglion cell can have multiple inputs into it, and especially if you move off the fovea, then you're going to see that happen a lot more. In fact, if we see, like, this is supposed to represent more of what's happening in the periphery outside here, and then we can see the organization more of like what we saw before, with a bipolar cell here and a ganglion cell here and a one-to-one correspondence, much like we saw here. And it says that is happening here in the fovea. So, all right. So that's this correspondence here. These things here, which they've described as being sort of a hat-like structure here, where they have these graphic depictions here, are useful in terms of being able to help us understand how the how the retina is responding to, or how different portions of the retina are responding to a stimulus. So what I want to do is I want to kind of walk through this a little bit here. So if we look at this kind of depiction that we have here, what helps to think about is first the setup. What's happening in this case is that you have a microelectrode that's being placed into an on-center ganglion cell, and that's what you're recording from. And so you have that hooked up to a device, and you're tracking to see whether do we have more or less voltage being created, as we shine a light over here or over here. And what this is trying to depict is that for an on-center ganglion cell, if you shine light onto this center area here, then what that'll do is that'll stimulate it and make it become more activated. And the way that it represents it is that depolarization is shown here on the z-axis. So up means depolarization. You can see the color changes here representing the different levels of depolarization. And then the x and y-axis are simply where the stimulus is being placed on the retina, on the visual field. So if you shine the light right here, for example, that would correspond maybe to like this region right here. If you move your light over and you shone it over here, for example, then that would correspond to somewhere over here. And this is showing on a XY field, so it's like a sheet, which is like our receptive field is like a sheet. And our retina, if you spread it out, is like a sheet. And so what we have here is we're representing what this particular ganglion cell sees. And how does it respond as the stimulus is being presented and moved across a part of this sheet? And the point of this is to help depict that there's a circular type of pattern here, where you have sort of the center area here with a massive depolarization here, and then a surround out here where there's actually some hyperpolarization that occurs, and that it's suppressed. And what this is supposed to represent, basically, is that if you shine light in the middle, right here that we were talking about, great, this on-center ganglion cell will then depolarize more. But if you move the light out here into this area here, and you shine light here, what will happen is that these off-center cells will end up communicating to these on-center ganglion cells and suppress the activity there. So there's a communication that happens between these different cells in these different areas that help to shape what this particular ganglion cell sees. So that's the idea behind this image. And here is an off-center ganglion cell. This means that when you shine a light here, that the cell becomes hyperpolarized, doesn't fire as much, and then as you move out into the surround, it becomes depolarized more, and so the hat is inverted. So that's just kind of cool. Dig into this and think about this a little bit as you're going through here, because I think if this starts to connect and make sense to you, you're getting the bigger picture. But let's break it down a little more and see if we can do this. And so what I'm going to try to do is just focus on some on and some off center cells here and see if we can actually, work with some simple schematics to make it a little easier to break down. And what I'm going to do is represent part of the retina here in these concentric circles here of receptive fields. And so these gray spots are representing photoreceptors. And then what we're looking at is that these photoreceptors are feeding into ganglion cells. And so what this receptive field is trying to represent is what the receptive field is for a particular ganglion cell. And so what we have basically is something like, let's say it's like an on-center ganglion cell here. And so we get some input here and some input from over here. So gray spots over here in the off surround. And then some of those spots here for the on-center. Off surround here, and some of these spots here for on-center. And so there's a lot more of those receptors spread out throughout here. And so you can kind of see how I've depicted it here, where we've got like a lot more of these different types of on and off center ones. But let's go back here and kind of keep it a little more simple, and we can take a look at that. Okay, so fine. What is going on here? So I took the original image that we were working with, and I kind of cropped it a bit to work to modify it so that we could actually track this a little bit more easily. So that image is actually a little further ahead. Don't panic. We'll get back to it. What I wanted to do is basically show the responsiveness of our ganglion cell and how it actually is responding to to this. So let's go ahead and just draw real quick our ganglion cell. And I'm just going to write G here. And so this ganglion cell is an on-center. I'm not very fast at this, but that's probably a smart thing. That's on-center, and meaning that it's, so this is the on-center area of this. So this ganglion cell is getting input from bipolar cells that are here. Good Lord. This is very professional. There's probably a better way to do this. Bipolar cells here. And these bipolar cells are connected to photoreceptors. So let's draw another bipolar cell. And this process, I'm illustrating 3 here, but there's more. So bipolar cell. That's tucked in and feeds into here. So you can envision that there are these bipolar cells that are connected to photoreceptors represented in gray, and that they're carrying their information on to this ganglion cell. So there's a convergence of information as it's feeding into this ganglion cell. And these are, there's an on center, an off surround. So what we're looking at then is that we're asking, if we put a microelectrode here, and we ask, well, what does and how does this ganglion cell respond to a light stimulus that's being presented and shown across here? What is it that's going to happen to that firing of the ganglion cell? And so if we do that then, and let's go ahead and change the color here to yellow. And we start off, if we shine the light out, let's say out over here. Oh dear, we can see that. Let's try that again, pen color. We'll go for green. Okay, so we shine the light out here, right? and we're shining it here. Well, this is outside of the receptive field of this ganglion cell. So even though we're on the retina, some other ganglion cell might pick up on it, but not this guy. And so there's nothing that's going to happen. We have a baseline activity here. And so this is our first sort of stage where we're sort of outside of that area. And so I'm just going to write 1 as a number 1 here. And then what we do is that we move that light stimulus and we scoot it over and we happen to hit, let's say we shine it on these guys. Like we shine it here, and maybe we shine it, it's a bigger sort of bar of light or something, and we're kind of shining it here. So we're activating these bipolar cells that are off bipolar cells. So that means you shine light on that photoreceptor, then that means that suppresses the activity of the bipolar cells, and that in turn suppresses the activity or has less activation to pass on to the ganglion cells. And so what happens then is that the cells become hyperpolarized relative to the resting state. And so number 2 that's being shown here is that kind of condition. Then what happens is that we move this light and we say, okay, let's move it over from, we used to have it here, then we moved it here. Let's go ahead and move the light over here and shine only on the center, not on this other stuff, not on here. And I'm just going to label this number 3, or step 3. And now that we've moved that light over into the center, what ends up happening is that we end up stimulating the cell and stimulating the ganglion cell. So that's number 3 here. And as we move this light across here from 2 to 3, if we stopped like right here and had the bar of light sitting right in between the two, so we're kind of hitting the off and the on at the same amount, that would be right at this point. I'll call this 2.5. So okay, fine. So what we're looking at then, and what this is, what I'm trying to illustrate is that this movement of light across the receptive field of this ganglion cell changes how it fires and how it behaves. And so we're sort of seeing how the retina, if we were just to look at the retina, it would just be this huge field of photoreceptors and unmarked bipolar cells and ganglion cells behind it, and we wouldn't know what is what. And meaning we wouldn't know what the receptive fields are and how it's organized and everything. But by hacking in and listening to the electrophysiological responses of the ganglion cells of bipolar cells, we learn how these different photoreceptors are wired to feed into bipolar cells and then in turn ganglion cells, such that the receptive field of the retina is organized into these smaller sort of concentric circular receptive fields. So, and I would probably step back and just say the whole retina is described, you can think of it as a sensory field. And then within that, there are these receptive fields. And receptive fields is a term that you'll hear a lot in sensory physiology and in neuroscience, describing how it is that this ganglion cell receives information, the field of reception that this particular cell has. And so in this case, in a visual modality, we can kind of imagine it in this sort of two-dimensional space that we have here. So this is how we have it set up in this case. Going back then, we can see on-center cells here, off-center cells here, and we're talking about how they communicate. And if we looked at the entire receptive field here, we could see that this is, and we're shining the light all the way around the retina. There's a two-dimensional appearance to it, so that there's an off surround and an on-center. So this is depicting more of those photoreceptors scattered throughout the receptive field of this particular ganglion cell that we drew before. This is the same kind of picture here. And this is just to show that these are, I kind of put them one next to each other here. So we can imagine like, okay, well, this is helping us be able to see that multiple ganglion cells, ganglion cell 1, ganglion cell 2, ganglion cell 3, would have this type of organization. So it's not a complex picture here, but or a big point, but it still is one that can be helpful. So let me just go ahead and say that we have this ganglion cell. I'll just call it ganglion cell number 1, ganglion cell number 2, and then ganglion cell number 3. And it's receiving input through bipolar cells that then are feeding into it. And there's lots of these bipolar cells here, here, here. They're all feeding into creating. So ganglion cell number 1 sees the visual space right here. You shine light right there, then this ganglion cell is going to respond. If you shine light over here, like you shine a light right here, this ganglion cell would respond, and this one would not. It wouldn't see it because it's outside of that receptive field for ganglion cell number 1. And so these kind of feed into, and I'll kind of do a sort of showing how they funnel into this information for these different ganglion cells. And so what I've been doing is I've been focusing on, or I talked about how we have on center, off surround, receptive fields. What's kind of cool though is that these overlap. So an off surround actually corresponds to an on center ganglion cell region here. What I mean by that is that these may be on center, off surround ganglion cells right here, but just adjacent to it, we have off center, on surround ones. And so here, what we have is this receptive field here is an off center, and this is an on surround just outside of it. And so, let's see, I can, this picture we can come back and we'll see it in another image to try to help clarify it. But there's overlapping receptive fields that happen all across the retina. And so there's multiple representations of the same area within the retina. And that helps us to be able to appreciate that we're not relying on a single ganglion cell to represent a certain square footage of our retina and our visual field. Instead, we have multiple ganglion cells that respond differently. Some are on center, off surround, some are off center, on surround. And so by having this kind of multiple coding of the visual stimuli that hit our retina, we're able to add redundancy into the system in terms of being able to provide a stimulus response to a stimulus. But we also are able to add that redundancy helps us to be able to ensure that we have fidelity, but also by having a different type of response, not everything's on, not everything's off center, we're also able to ensure that we're covering the diversity of types of responses or stimuli that we can sometimes see. And so it adds to the fidelity overall of our visual experience. Okay, so this is kind of returning back to that image that we had before, where we have on center and off surround, and looking at this kind of depiction of our receptive field here. So again, we'll go back and we'll look at this at the beginning on Thursday, because I just want to make sure it's a clear one and that we have a chance for questions and answers on that. And this is our original image, and we'll revisit that as well. But it's the same concept that we were just talking about. Okay, so the next couple of slides help us to be able to look at this and be able to, and it's another representation of some of those types of concepts I was just illustrating. And so it feels a little redundant, then that's great, but just trying to make sure we're getting as many kinds of snapshots of this thing here. So ganglion cell receptive fields are ones that they have on-center and off-center cells. And these respond to differences in illumination, as we had talked about before. And so an off-center ganglion cell output, this is looking at sort of a little patch of the retina that a ganglion cell is picking up on. And then there's his center, and here's the surround. And then if there's no input happening if there's broad diffuse light being shown to it, you're stimulating both the center and the surround here. And so there's this modest amount of activity here. But if you shine the light only on the surround and not on the center, then what happens is that this becomes really active. And then if you shine dark light over this, then you can see that this kind of activity gets suppressed here. So it's not quite equal here and here. So there's some interesting differences in terms of response to different types of illumination. But that helps us to be able to see what we're seeing. And so if we think about not just having like a dark spot here, but actually moving an edge of light, which is sometimes more realistic, what you can see is this kind of responsiveness that occurs. Like first broad diffuse light here with some activity as we look at how the off center and on surround sort of battle it out with each other to do it. Here we have this dark edge here. So the light is only shining on this center part. It's an off-center ganglion cell, so there's suppression of activity. And then as this light dark edge moves across here, the off-center here becomes more dominant. And so it starts to fire more. And then ultimately spreads over that whole area. As we move that darkness across here, we can see that the activity of the on surround then becomes is able to have more of an influence. So, okay, fine, and this is actually just a larger picture of that. In addition to just the intensity of light, we also are able to detect differences in wavelength of light, and so that we can actually have red and green receptive fields. So this is, I think, kind of awesome because we're looking at red on and green off surrounds. So adding color as an overlay to our images here, we can see how this responsiveness of a red on, green off surround type of ganglion cell is constructed. So we're not only able to discriminate between the light intensity as it moves across the visual field, but also color changes and color differences as it moves across the visual field. And all of this is being processed in these small receptive fields that we have for these ganglion cells. And so we have multiple types of ganglion cells, and we'll talk about those differences in more detail. But magno and parvo and non-M, non-P cells are the major types of descriptions for them. And then we have these sort of color opponent ganglion cells that we have, which represent parvocellular non-M, non-P cells. So let's kind of dig into some of that and think about what these different types of cells that we have are. So retinal ganglion cells are they have a M and we have M and P cells and non-M, non-P cells. So this is sort of a simple depiction of an M-type ganglion cell and a P-type ganglion cell. The non-M, non-P type cells are out there as well, but these have the most distinct morphological differences here and help to kind of reflect what it is that they're actually doing. So not the M type are really good at having and detecting motion. So they tend to have really large receptive fields, larger cell bodies. They're sensitive to visual stimuli that change quickly over time, so they're able to adapt really quickly to a change in a visual stimulus. They have large receptive fields, and you can see that in part because of how they have their dendritic arborization for picking up the sensitive inputs. And then they're more sensitive to contrast. And so they respond with this transient burst of action potentials. So they're really fast. You shine a light, they start to fire, and then they quickly adapt. And this allows them to be able to have that kind of sensitivity to visual stimuli that changes quickly over time. And this makes about 5% of all the retinal ganglion cells that we have. The P-type ones here represent about 90% of our population of the retinal ganglion cells. They have smaller cell bodies and smaller receptive fields here, and they're sensitive to high-frequency spatial stimuli. So they're less sensitive to contrast, but they respond with more sustained type of discharge of action potentials in their responsiveness. So the non-M, non-P type of cells that we have here make up the remaining 5% of these retinal ganglion cells. And these are, we don't know them as well, but they definitely are involved in color vision in terms of their function. Okay, so the ganglion cells and where they're positioned and what they're doing, where do they carry their information and how do they actually communicate? So we're looking at the retina kind of tucked in back here, and then we're looking at how we have these different three different dots here, which nicely correspond to magnocellular with a big dot, and then parvocellular, which is being depicted with a smaller dot, and then the non-M, non-P ones that are being depicted with a smaller dot as well. But we can track back these dots and look at how they're feeding into the part of the thalamus called the lateral geniculate nucleus. So let me take you over to this image, and let's kind of just look at it on an anatomical level that feels familiar, and then we'll return back to over here. So light shines in, hits the retina. Then we talked about photoreceptors, bipolar cells, ganglion cells. Ganglion cells reside then. Let's go ahead and use the laser pointer option here. It's kind of nice. So ganglion cells that are over here then carry their information back to the thalamus. And then the thalamus and the lateral geniculate nucleus, or LGN, is the part of the thalamus that we're looking at. The thalamus then in turn sends information back to the occipital lobe and the visual cortex, and the primary visual cortex in particular, located in the calcarean fissure of this area. And so if we were to dig into this and blow this up a little bit, we can see that the lateral geniculate nucleus, that little part of the thalamus here, has multiple different layers that are tucked in with each other, and that they have parvocellular and magnocellular layers as part of their inputs that are feeding into it. The big thing I just want you to appreciate first is like, okay, ganglion cell body is here, feeds back to the LGN. All right, that synapses onto neurons within the LGN, and then those neurons then project back to the occipital lobe, right? So these are the retinal ganglion cells, axons feeding back, and then they synapse onto like this neuron here, and then that neuron then carries its information back to the primary visual cortex. So now that we kind of appreciate, okay, first leg of the journey, second leg of the journey, what we can do is kind of talk about how this is organized a bit, and how it actually breaks down into the types of retinal ganglion cells that we have. So these magnocellular cells that are more responsive to motion, fast transient responses to visual stimuli, etc., are being depicted here, and they feed in to, in this eye, for example, and they feed into layers 1 and 2 over here. And so magnocellular one on the ipsilateral side. And what I mean by that is, if we look at this, two eyes here, midline crosses along in here, just to make sure we're all on the same page with the anatomy here. So this is the midline. This is known as, this is the left and the right side. So if you have a stimulus that's kind of hitting here, for example, on this side of the visual field, then you can refer to the structures as being on the same side of the midline as being ipsilateral. And so they're on the same side as the point of reference that you're talking about. So if you're hitting this particular part of the retina, for example, like this guy, what happens is that you take your information and it ends up, for the way the retina works, is that that information is then carried over and it feeds into layer 2 of the lateral geniculate nucleus. So this is getting ipsilateral information. Magnocellular cells over here on this side, let's see, the stimulus happens to hit over here and hit this particular retinal ganglion cell. That carries information back down. This crosses over the midline and feeds into the magnocellular layer that we have here, layer 1. So you have layer 1 and 2 receiving magnocellular input. Layer 2 is getting it from the ipsilateral side. Layer 1 is getting it from the contralateral side over here. And so usually what I do if I'm trying to depict this and remember this is I'll just draw 2 circles, or 2 hemi-retina, I guess. And then with a line in the middle. And then I'll just draw an X here and here, and then let me change the pen color real quick to like a blue. And then to represent the connectivity of these other part of the retina, I'll kind of draw another different color. This helps me to remember which cross the midline and which ones don't. Because what we're looking at here just to try to help here, is that we're looking at what's a representation of the retina. And the part of the retina that's closest to the nose, so I'll draw the nose here, the nasal part of the retina, actually these retinal ganglion cells then project and cross over the midline. And whereas if we have this part of the retina that's over here, we'll carry its information over and we'll keep the information on the same side. So we're kind of looking at, in this depiction here, we're looking at, this is the light that's happening to hit this outer area right here. And so it stays on the blue, sort of same it gets processed on the same side of it. And then we're looking at light that happens to hit over here and stimulate here, and it crosses over and feeds into the lateral geniculate nucleus here. So that's the type of organization that we're looking at. So this image here has a lot kind of going on to it. So let's kind of clear it real quick and take a look at it. So what I drew for you guys is I basically said, okay, we've got, and the thing that I was telling you about, the first thing I was telling you about was that we have this magnocellular input, like from this here, that feeds into layer 2, and magnocellular input over here that feeds into layer 1. And they cross over. And what I was adding to it was was that there is this kind of way that the retina is made, and we'll go ahead and split this in the middle. And so that whatever's on the temporal side of the retina over here, that information ends up kind of staying on the same side and processed by the LGN on this side. So both magnocellular and parvocellular inputs get sent over here. Input that's sent over, and I'll change the pen color to red. Input that strikes over here ends up getting shunted over to the opposite LGN over there. The same kind of thing happens over here, where the temporal side gets processed on the same side of the by the LGN on the same side of the eye and the same side of the midline. And then any of the light that happens hits on the nasal side of the retina here, then gets shunted over in this direction. So I hope that helps clarify that. We can just touch on it when we meet up on Thursday and make sure that's okay. So I focused a lot on the magnocellular cells, depicting that. Parvocellular input then feeds into layers 3, 4, 5, and 6 of the LGN, and some are from the ipsilateral side and some are from the contralateral side. So one thing to think about is that your lateral geniculate nucleus here, that's tucked away into your thalamus here, is receiving inputs from both sides of the visual field, 2 different eyes that are carrying that information. So how do we remember this? Well, this is kind of a simple way of kind of breaking this down, where we look at in these different layers of the LGN, 1 through 6, we have contralateral and ipsilateral eye being represented. So which one's contralateral, which one's ipsilateral, how is it feeding into? So contralateral input feeds into layer 6, ipsilateral input into layer 5. And then if we're breaking it down into the types of ganglion cells that feed into these different layers, we have it as P-type or M-type, or non-M, non-P. And then finally we have, and that helps to characterize how the LGN is responding to it. So what we have that is magnocellulars in layer 1 and 2, and then parvocellulars in layers 3, 4, 5, and 6. And then what we have is these non-M, non-P inputs that are coming in as well, that are these coniocellular layers that are sort of in between these other layers that are being depicted of 1 through 6. So this is a useful one to kind of help kind of structure your learning and thinking about how to sort of formalize and where everything is. So an easy thing to kind of use for memorization. Okay. So let's add complexity to this, because that's what we do here. And what we're looking at then is how the lateral geniculate nucleus that's present is feeding into the primary visual cortex. And in the previous pictures, we've been sort of saying, like, okay, yeah, LGN feeds in here to the primary visual cortex. But what's really kind of interesting is that this whole retina here is represented and has a spatial representation in terms of how the axons are even projected back from the LGN back to the visual cortex. Let's take a look at what I mean by that statement. The LGN here comes back and feeds into different areas of your visual cortex. So you're mapping basically your retina on the back of your brain over here. And the way that mapping happens is that some of those projections actually go and are present a little closer in to where we're the lateral ventricles, and then some are a little further out and a little more superficial along the calcarine fissure here. So this is usually around where the calcarine fissure is. And so you have this kind of representation. In fact, if you shine a grid onto the stimulus, onto the retina, and shine them onto there, and then you looked at the activity that was occurring in terms of the cells that were responding to it in the back of the brain here, you would find a representation, a map of your retina being shown on in the back of the brain here. So your fovea is actually highly represented in the back here, and then as you move outwards, your periphery is represented. And your fovea actually has a much higher amount of representation than your periphery does, and that's in part so that you can be able to process the more detailed type of input that comes in from the foveal portions of your eye. The other thing that's kind of interesting is not only are you have this kind of representation here and a lot of representation of the fovea versus the periphery, but this anatomical representation of these this inputs that go from the lateral geniculate nucleus, this map that's here, helps to correspond to different areas of each eye. So we have green and blue showing different regions of the left eye and the right eye. And these are being carried back by the lateral geniculate nucleus. And depending on where you would have damage along these different structures can influence how you actually see and how you actually have the changes in your visual field. And so to help illustrate this, there's a separate image that I loaded. It's called loaded onto the blackboard, but I integrated into this structure here. So if you take a look at that PowerPoint slide, you'll see it here. But the point of it is basically showing how this wiring is between the retina that we have here and how it projects back to the visual cortex. So as you have a light, let's say that we have these different types of colors here. And then say that the left side of your, or left eye over here, your left visual field, and then the right lower quadrant of it. So the right lower quadrant of your visual field. Well, when you shine that light into the eye, it goes through the lens and gets flipped. So so that corresponds to this upper part right here. And then that in turn gets carried back to, along by retinal ganglion cells, back to the lateral geniculate nucleus, synapses, and then feeds back to optic radiations, in this case, to the visual cortex. And so damage here, at this particular point, would cause damage to this quadrant here. And so that's kind of a representation of what we're seeing there. So okay, that's really neat. We kind of see how that works. But I want to go and actually let's look at this color, this guy right here. What's running alongside with this? If we trace it back, we see that it actually corresponds to the other eye in its lower right lower quadrant here. And so what this is telling us basically is that each eye has been wired such that we're actually carrying that representation along the same anatomical area of our optic radiations here. So even though they're coming in from separate eyes, the organization of the wiring is such that we're able to represent these right lower quadrants of the visual field in the same area of your eye. Same area of your brain. So you have two representations there of the right lower quadrant, one from your left eye and one from your right eye. And those are both being carried back around the same sort of portion of these optic radiations that are feeding back. Meyer's loop, another area here, is then showing this type of responsiveness that we have from the right upper quadrant. So that's the feedback that we're kind of seeing here. And the other thing that you can sort of appreciate then is that over here, we're looking at these two different regions here. So we have right lower, left lower part here, and so that's being here, and we have and this guy over here. And then if we look at what's happening on this part, it's being carried over and processed by this other side of the retina here. So your right part of your visual field is being carried back by this portion of the retina on the left eye. The right part of your visual field is being carried by your nasal part of this retina here and feeding back into the brain towards the LGN. And what's interesting is that then what all the wiring that we have here is responsible for processing input from here and from here. So if you have a tumor or something that knocks out your visual field here, you're going to get a different pattern of damage occurring to your visual field than if you have a damage occurring here or here in your visual field. So stroke can along the visual pathway can sometimes tell you about where the damage is occurring if you're able to analyze the type of responsiveness that the person's actually having. Okay, so we spent a lot of time talking about how the LGN's connected to the cortex, and now let's start looking at how that visual cortex and the receptive fields work. We'll begin that in part 2 of our series. I'm going to stop it here, give us a little break before moving into the next phase.